I'm Professor Rosie Redfield, and this short video is to give you a sample of what students will learn in my free online course, Useful Genetics. DNA fingerprinting is constantly in the news. It's used to identify and convict criminals, to reveal family relationships, and to free people who've been wrongly convicted. Why does DNA make such good evidence? Well, it's mainly because we all leave DNA behind us everywhere we go. This DNA is very stable. It can even be extracted from fossils, and it can be studied in tiny amounts, even in complex or dirty mixtures. Most importantly, DNA sequence differences uniquely identify every individual. The differences are even better than fingerprints. We found that the best way to use these differences for identification is with a carefully chosen set of differences called markers. Each marker comes in many different versions so that there's not millions or billions, but trillions of possible combinations. Each person has two sets of these markers, one from their father and one from their mother. And when they have children, because the markers are on different chromosomes, each child gets a shuffled combination of these markers, a set from each parent. Each different sibling has a different set. The only identical siblings are identical twins. But family relationships are still easy to see because each child's versions must come from their parents. The differences are easy to analyze because markers were chosen to be places where the length of the DNA varies because there's different numbers of repeats. This means that the analysis can be automated by machines that are not only very efficient, but also very reliable, giving very reproducible results. So why then is DNA fingerprinting still controversial? Well, the truth is that the problem isn't in the machines, the problems are in the people. Every DNA sample passes through many hands on its way from the crime scene to the courtroom. Attention is now focused on maintaining careful control in what's called the chain of custody. So this was just a tiny sample of what you'll learn in Useful Genetics. If you haven't already, you can sign up for free at Coursera.org. I hope to see you there.